Let's dive right into this explainer, where we're going to zoom all the way in on how science has managed to organize every single living thing on our planet. We're talking moving from the macroscopic stuff you can see down to the microscopic stuff you can't. If you've ever looked around at just the sheer, overwhelming variety of life on Earth and thought, how do we even begin to make sense of all this? Well, you're in exactly the right place. Today, we're cracking the code of biological classification.
You know, back in the day, folks like the ancient philosopher Aristotle basically just looked at plants and animals and tried to group them by what they could literally see with the naked eye. For plants, he just sorted them by height: herbs, shrubs, and trees. Simple, right? And for animals, he asked questions that seem super basic to us now, but were totally logical for his time. Things like, do they live on land, in the air, or in the water? And, my personal favorite, do they have red blood or not? It was a really noble start for sure, but as our tools got better, we quickly realized the naked eye was totally missing an entire invisible universe.
So, here's our mental map for today's journey. We'll start with the evolution of classification, then hit Whitaker's 5 kingdoms, move into Kingdom Monera to meet the prokaryotic pioneers, check out extreme versus true bacteria, and finally wrap up with Protista, the very first eukaryotes.
All right, kicking things off with Section 1, the evolution of classification.
Okay, so the way we classify life, it definitely didn't just happen overnight. It was literally a centuries-long awakening. Our scientific lens basically kept zooming in further and further over time. We started with Carolus Linnaeus back in 1758, who gave us a super simple two-kingdom system, Plantae and Animalia. But then, microscopes came onto the scene. And suddenly, scientists like Ernst Haeckel and Herbert Copeland were like, wait a second, we're completely ignoring all these single-celled organisms. That pushed us to 3, and then 4 kingdoms, and that eventually brings us to the monumental shifts by R.H. Whitaker and later Carl Woese, who realized this microscopic world was so incredibly complex, it actually needed its own distinct domains.
Which brings us to Section 2, Whitaker's 5 kingdoms.
This massive shift in 1969 gave us the famous system that is still widely accepted and taught everywhere today. Fun fact, before Whitaker, fungi were actually just awkwardly lumped in with plants. Crazy, right? But Whitaker didn't just guess to fix this. He used a really strict set of criteria to methodically separate life out. He basically asked, is the cell primitive or advanced? Is the organism made of just one cell or is it multicellular? Does it make its own food like a plant or rely on eating other things like an animal? And finally, how are they all linked by evolution? By rigidly applying these rules, he gave us the 5 kingdoms: Monera, Protista, Fungi, Plantae, and Animalia.
Moving right along to Section 3, Kingdom Monera, our prokaryotic pioneers. If we want to understand the most ancient, single-celled organisms on Earth, it all starts with one defining trait. Every single member of Kingdom Monera is a prokaryote. That means they operate without a central nucleus or any membrane-bound organelles. Their DNA is just lying naked right there in the cytoplasm. But, don't let that primitive structure fool you for a second. These cells are the fundamental, rugged pioneers of life. They can survive almost anything you throw at them.
And even without all those complex organelles inside, these single-celled pioneers actually come in a visually stunning catalog of shapes. In fact, scientists largely group them just by what they look like under the microscope. You've got the spherical ones called coccus, the rod-shaped ones called bacillus, spiral-shaped ones known as spirillum, and my personal favorite, the vibrio, which look remarkably like tiny microscopic commas.
Now, when it comes to actual survival, bacteria are incredibly diverse in how they get their meals. On one hand, we have the autotrophs. These are these self-feeders. They can literally synthesize their own food using light energy, kind of like plants do, or they can even use chemical energy they extract right out of their environment. On the flip side are the heterotrophs. These act as decomposers, breaking down dead matter, parasites living off a host, or even symbiotic partners that form complex relationships to survive.
Let's jump into Section 4, Extreme and True Bacteria.
So as scientists kept studying monerans, they eventually realized, okay, we actually have to split this kingdom into 2 drastically different groups, mainly based on how they survive. First up, we have the Archaebacteria. These are the ancient, incredibly tough organisms with specialized cell walls that let them thrive in totally extreme places. We're talking boiling hot springs, highly salty environments, or even deep inside the guts of cows where they produce methane gas. Contrast that with the Eubacteria. These are the true bacteria that live all around us, basically the ones you automatically think of when you hear the word bacteria.
A perfect example of these true bacteria are cyanobacteria.
Now, for the longest time, people would look at ponds, see this green scum, and just call it blue-green algae. But when scientists finally zoomed in and looked closely at the cells, they realized, wait a minute, there's no nucleus here. These aren't algae at all, they're actually photosynthetic bacteria, capable of massively multiplying in water to create those huge algal blooms we see.
One absolutely fascinating trick some cyanobacteria can do is a process called nitrogen fixation. Think of our nitrogen-packed atmosphere kind of like a raw potato. Living things just can't digest it. But these bacteria have special cells called heterocysts that essentially cook that raw atmospheric nitrogen into a usable chemical form. It's like turning that raw potato into French fries. Suddenly the plant and ultimately the rest of the food chain can actually digest it and use it to grow. It's brilliant.
And just when you start to think you've figured out the rules of Kingdom Monera, you run into mycoplasma. These are literally the smallest living cells on the entire planet, and they stubbornly break all the bacterial rules by surviving entirely without a cell wall. Oh, and if you take away their oxygen, totally fine. No problem at all. They can survive completely without it, often acting as really sneaky pathogens causing diseases in both plants and animals.
All right, Section 5, Protista, the first eukaryotes.
Stepping up the evolutionary ladder here brings us to a wild and hugely diverse group of single-celled organisms that bring a major upgrade to their cell structure. Every single organism in Kingdom Protista is eukaryotic. So unlike those primitive bacteria we just talked about, these cells finally have a proper, membrane-bound nucleus to protect their DNA. It acts like a true control center. They also feature complex organelles, things like mitochondria and Golgi bodies. It's literally like moving from a one-room shack into a highly organized multi-room factory.
Let's check out a mesmerizing group within Protista called the chrysophytes, and specifically the diatoms. These guys are the chief producers of the oceans, just floating passively out there in the water currents. But their most mind-blowing feature is absolutely their cell wall. They essentially live inside indestructible glass houses. Their walls are infused with silica, and they fit perfectly together in two overlapping shells exactly like a tiny microscopic soapbox.
And here's the crazy part. Because those glass shells are nearly indestructible, they don't just decompose when the cell dies. Instead, over millions of years, trillions of these dead diatoms sink to the ocean floor, creating massive, gritty deposits known as diatomaceous earth. It's an incredible full-circle moment where biology straight up meets geology. This microscopic glass powder is actually harvested by us and used in our daily lives for everything from polishing metals to filtering oils and syrups. Who knew, right?
Another spectacular, though admittedly dangerous, member of Protista are the dinoflagellates. These marine organisms have two little tails, or flagella, that help them move around. But under the right conditions, they can multiply at an explosive rate. When this happens, they can literally turn the entire surface of the ocean red, creating a phenomenon known as red tides. It's an awesome sight for sure, but they also release potent toxins that can cause massive die-offs of other aquatic life.
Finally, we have the euglenoids. These are the ultimate evolutionary fence-sitters. Remember earlier how we said plants make their own food and animals hunt? Well, euglenoids do both. Instead of a rigid cell wall, they're wrapped in this flexible protein layer called a pellicle. This lets them gracefully swim into the sunlight where they act exactly like plants, using chlorophyll to photosynthesize. But if you plunge them into darkness, they instantly switch their behavior. They literally start acting like predatory animals, hunting down smaller microbes for food. Talk about having the best of both worlds.
So we've really only just scratched the surface of the single-celled universe today, moving from the simplest, toughest bacteria to the highly adaptable protists. But it leaves you thinking, if a single, totally invisible cell can build itself a glass box, survive boiling acid, or switch between being a plant and a predator on a whim, what kind of bizarre, spectacular quirks must exist out there in the massive multicellular kingdoms of fungi, plants, and animals? Well, that is a journey for our next explainer. Thanks so much for learning with me today.
Thank you so much for visiting my YouTube channel and my unofficial biology on Facebook page. Thank you.